the beta hemolysis on blood agar. So we make cultures with Staphylococci aureus on blood agar. The colonies are creamy colonies. You have written that. Inside the genus, except this cream pigment, some species release an yellow pigment, and this is saprophyticus. Staphylococci saprophyticus. It's resident on the mucous membranes and the skin of humans, and it is not real pathogen. For Staphylococci species, here I will ask you, find me broth and agars for Staphylococci, for example, at the end of the semester. This one is Staphylococci. This is MRSA screen agar for methicillin-resistant Staphylococci species. Only the resistant species that possess MEC-A gene are growing on this agar. The susceptible strains do not grow. Salt milk agar for stool examination, especially. The pigment is creamy again for Staphylococci aureus. Blood agar with beta hemolysis, with creamy colonies. Salt broth for Staphylococci, and fermentation of mannitol. You can add, of course, plasmacoagulase tests. I couldn't find it, but you know it, and plasma agglutination test. What about Salmonella species? Salmonella species are representatives of family of Enterobacteriaceae, or they are gram-negative enteric microorganisms. They grow on common media, on blood agars as well, but the special group of media for such bacteria are the differential media. We have here levin and desoxycholate citrate agar or apocolate citrate agar, on which media we can differentiate the lactose positive and lactose negative enteric bacteria. Salmonella species are lactose negative bacteria. So you should show me the levin as differential media, lactose negative colonies, apocolate citrate agar with lactose negative colonies, and the Kligler of Salmonella. There are two groups of Salmonella: producers of hydrogen sulfide and non-producers of hydrogen sulfide, and a special strain which is Salmonella typhi species with this black ring because of the hydrogen sulfide. The hydrogen sulfide producers resemble very much the Kligler of Protea species, as I explained you that last week, and we can differentiate Salmonella from Proteus using phenylalanine desaminase test or the FAD test. Proteus species are fat positive, while Salmonella species are fat negative. And non-hydrogen sulfide producers will have Kliegler like the Klebsiella species. No, this is not so good. Maybe this one. With lactose negative slant, glucose positive, gas positive, hydrogen sulfide negative. This is for Salmonella. And maybe you should say that the selenite broth is the enrichment media for food poisoning examination and for identification of enteric bacteria as well. What about Clostridium perfringens and Clostridium botulinum? They are, like Campylobacter of course, they are fastidious microorganisms. They need the richest blood agar, which is Zeissler agar, with 20% sheep erythrocytes. They are slow growing. The colonies appear after 48-72 hours of incubation. And for Clostridium perfringens, we can say that there is a double zone of hemolysis on blood agar, a beta and alpha hemolysis, while the colonies of Clostridium botulinum are semi-perspirate, without hemolytic zone. And what else? We spoke about Campylobacter uni last time, and now here are almost all of the media that you should know. Common agar for non-fastidious microorganisms, common agar with a caput medusa colony of Bacillus anthracis. Here is the cotton deposit, where was it, of Bacillus anthracis in glucose broth. Here the pigmented colonies of Staphylococci saprophyticus, Pseudomonas aeruginosa with green rough colonies, Proteus species like waves here. All these are common agars. This is Müller-Hinton agar. These three plates here for disc diffusion method, for antibiograms, for non-fastidious microorganisms. The fastidious are tested on blood agar, or chocolate agar, or Levinthal agar. Oh, yes. MRSA screen agar, I spoke about that. Alkaline agar and TCBS agars are for Vibrios, especially Vibrio cholera. This rice or cornmeal agar is for chlamydospores of Candida albicans and other fungi. These are chromic agars for Candida. The different Candida species grow with different color of the colonies. This is for preliminary diagnosis only. Here is the salt milk agar. We spoke about that. This is Levinthal, very much resembling the common agar and the alkaline agar and the rice agar as well. For fastidious microorganisms, for microanaerophiles, is this green pouch. Such are Haemophilus species and Neisseria species. And I shall continue with the blood agars. The poorest in red cells is the common blood agar with alpha hemolysis, alpha hemolytic colonies. The Optohin test that distinguishes Streptococci pneumonia from other alpha hemolytic or viridans streptococci. Here you have beta hemolysis and the bacitracin test for the susceptible group A Pyogenes species, and the CAMP test positive with this triangular phenomenon here for group B streptococci. You have E. coli hemolytic strain with beta hemolysis on blood agar, gray colonies with beta hemolysis. You have the satellite culture for Haemophilus influenzae for the two factors hemin and phosphopyridine nucleotide, chocolate agar for Haemophilus and Neisseria species, Bordetella for Bordetella pertussis, the causative agent of pertussis, and we spoke about the differential media, lactose positive colonies of E. coli, dark blue, lactose positive Klebsiella species on Levin but after the 24th hour because of the capsule. The lactose negative colonies on desoxycholate with dark center for hydrogen sulfide producers, which are Protea species and some Salmonella species, and the red colonies of lactose positive enteric bacteria on desoxy or apopylene citrate agar. Any questions?
Can you give any information about
About
how can we
I
use it?
gave you several times explanation about the practical exam. I'll ask you, find me here all the media for Staphylococci species, for example, and for the tests, or for the enteric bacteria, or for Bordetella pertussis, or find me any isolation or biochemical test concerning Klebsiella species or hydrogen sulfide producer, or I'll tell you, for example, Proteus mirabilis. You should know that there are two Proteus species,
Identification.
mirabilis and vulgaris, that are, and I'll give you preparation to look and to explain me. What are you seeing?
Identification.
Cells, how many cells, which cells can you recognize? Are there any microorganisms or not? The morphology of microorganisms, cocci, beans, or rods, or curved, how they are situated in pairs, in chains, in groups, extracellularly, intracellularly, and so on. Are there any acid-fast bacteria in your preparation? And these things. And of course, I'll ask you for all the methods of staining, simple and differential, that you don't know yet.
You ready?
So, be quiet, sit around the Petri dishes, and revise. How come 3-4 minutes? Do you have questions?